Joining us today on Superheroes of Science, we're excited to welcome Dr. Tiffany Lyle, Assistant Professor of Veterinary Pathology at Purdue University. So welcome, Tiffany.
Thank you. It is a pleasure to be here today, and I'm so excited to share my scientific journey and also to share a bit about the science that we do in my laboratory. So I'm going to share a presentation with you all. All right, and I am going to walk you all through the comparative pathology of the blood-brain barrier, but I'll start by sharing how I got to sitting in this chair. So I received my bachelor's degree and my veterinary degree from the University of Georgia in Athens, Georgia. Then actually came up here to Purdue and did my residency in anatomic pathology. Went out to the National Cancer Institute just outside of Washington, D.C. and did my PhD research, which really blossomed my interest to continue to pursue research for my career. And then I started on faculty here at Purdue. So what do I do every day? What do all those degrees allow me to do? So I actually do many different things throughout the day, all centered on research as my central mission. So I lead the comparative blood-brain barrier laboratory. I know that's a mouthful, and I'll walk you all through what that involves. I also work with investigators all over campus, anybody who's using animals in their studies as a veterinarian and as a pathologist, and able to walking through their studies and work with them in those and their use of those animals. I also have a wonderful opportunity to teach both pathology residents and veterinary students. I teach histopathology, so just basically teaching residents how to interpret complex cases. And then I teach veterinary students all about cancer, all of the molecular nuts and bolts involved in cancer biology. So I also have a wonderful opportunity to take students to Tokyo, Japan. I lead a study abroad program to Tokyo in the summer. And unfortunately, we didn't get to run it this year due to COVID-19, but we look forward to going back as soon as we can. It's just a wonderful opportunity for our students, any student enrolled at Purdue is able to attend. Participate in that program. So let's get into the blood-brain barrier. So what is the blood-brain barrier? So the blood-brain barrier is a complex vascular structure that's actually just in the brain. So it's one of the most tightest structures in the brain. So when we think about vasculature, we think about veins and arteries most often, but there's a really small vascular network that's actually called capillaries. And capillaries are the end stage, and when we think about that arterial network of taking blood to a tissue, and that's the smallest structure, that's the end of the road. So it's composed of endothelial cells. So endothelial cells make up the intersection. It has a basement membrane here in yellow, pericytes that are embedded in these in the basement membrane, astrocyte infeed, which make up the outermost layer. So together, all of these layers work to prevent any sort of toxins or nasty chemicals, drugs, anything that shouldn't be in the brain from getting into the brain. So that's why that barrier is super important in protecting our brain, so that we can continue to be who we are, so that we can continue, I can continue to be Tiffany, and you can continue to be whoever you are. And these capillaries actually protect the brain from any sort of invasion. However, in disease, particularly in cancers that grow in the brain, this barrier can get damaged. So that's the why of our lab. We're interested in understanding cancers that migrate to the brain, most notably actually lung cancer, particularly non-small cell lung cancer. So non-small cell lung cancer is the most common type of lung cancer, involving actually 85% of lung cancer cases. There's been in 2019 alone, there were over 200,000 new cases and over 142,000 deaths from lung cancer. With that, 56% of the lung cancer patients actually present with metastasis, in that these tumors from the lung have been identified in other tissues like liver, also identified in bone, and especially the brain. So we're interested in finding new ways to actually address and treat those metastatic tumors, particularly those in the brain, because once they get to the brain, they're really going to impact the survival of patients and impact who they are. As I mentioned, once we get invasion into the brain, we lose our sense of self. So I'm going to go over our model here. So how do we actually evaluate the brain metastasis? So what we do is here you're looking at an ultrasound of the heart of a mouse and the left ventricle. So these animals are under anesthesia, and while they're sleeping, they cannot feel any pain as the needle delivering the cells is injected into the left ventricle. So after this time period that they're under anesthesia, we wake them up and then are able to image them actually and see the cells as they migrate through the body. So here we can see the cells, particularly the highest number of cells here in orange in the brain, and we also get some migration of cells into the kidney and other organs in the body. But we're most interested in those in the brain. So then these cells are able to colonize the brain for about 6 weeks, and then within 6 weeks, we can see actually these tumors growing here within the brain tissue. So we can evaluate them and look at them more closely to see what damage has occurred and what we can use to target the brain. So I would like to show our most striking data, and that is in those astrocyte infeed. I mentioned in the diagram that beginning the astrocyte infeed make out the outermost layer of the blood-brain barrier. And here in the upper corner, you can see the astrocytes here again, that making up that outer layer. So in these images, we're looking at brain from a normal mouse that had no tumors involved, the brain around the tumor region, and then here the tumor, which is traced in white. And in all of these images, I've labeled the astrocyte infeed. Again, these blue feet here, they're actually labeled green. I've labeled the endothelial cells in red, and together, the green and red make yellow. So in the control brain, we can see that these capillaries are all yellow. So the astrocyte infeed and endothelial cells are on top of each other as they're supposed to be. And when we look at the brain around the tumor, we can see a little bit of red creeping out, but by and large, similar to what we see in the control, that we still have that combination, that co-localization, that yellow color. But when we look at the tumor here, so the tumor again is traced in white, and it's all this here in the center, all we see is red. We've lost all of that green staining, so that means that those astrocyte infeed are lost. We have damage to the blood-brain barrier. So this is important data for my laboratory to know that when we work with understanding drug delivery and understanding keeping that blood-brain barrier as a possibility for manipulation, that these astrocyte infeed are an area that we can look at. So again, looking at the control over here, all these yellow capillaries, and all of that is lost in that brain metastasis. So again, we can see that's how we can look at changes in the blood-brain barrier. So a little bit more about astrocyte infeed. They're an important membrane protein. They have control the flow of blood. They are important in controlling ions. They have a role in migration of other astrocytes. So astrocyte infeed have been known to have implications in other diseases, in Alzheimer's disease. They've been misplaced and decreased. In ALS, you have an increase overall in aquaporin-4. In Parkinson's disease, also loss of aquaporin-4, multiple sclerosis, MS. There's variations in aquaporin-4. You can get increase or decrease, traumatic brain injury, or breast cancer. So not only in lung cancer do we see changes in astrocyte infeed, there are other diseases that this has shown to play a role. So we can use what we know about other diseases to improve drug therapy. So ultimately, the overarching goal of my group is to improve drug therapy improve the targets of those drugs so that we can actually shrink tumors and ultimately improve not only survival, but the quality of life of patients. So thank you.
Wow.
Oh, wow.
Wow. And so is what you're doing with veterinary, with pathology, is that transferable? I mean, it sounds like it's kind of transferable over to people then.
Yeah, exactly. One of the core missions of my laboratory is one medicine, to think about how, as we model diseases in animals, that can then be reflected and take that data into human hospitals, into the clinic. So my laboratory is very unique in that we, many of the diseases and organ systems that we study not only are applicable in animals, but in humans as well.
I get tired of saying it. It seems like the more I interview scientists like yourself, the less I realize I knew. I never would have thought that things going on over on the vet side of things is actually helping people.
Yes. I mean, it's almost, you have to work hard to not be able to help animals and humans with what we're doing and the science that we uncover.
That is so cool. Let's start with how you got it. I know you gave us your career path, but I'm kind of curious, what was your passion?
So actually, as a child, my passion growing up, I always wanted to be a news anchor, believe it or not. And but as I got to kind of doing my own fake stories and investigating things, I really ended up getting to know a lot of scientists, whether through school or through friends of the family. And just asking them more questions got me more engaged in science and giving me more opportunities. So I just really, I think my passion for knowledge and my passion to learn new things is what got me so interested in research.
Oh, I love that.
What were some of the first research experiences that you had?
So actually, my very first science fair, actually when I was a high school student in the 9th grade, I was able to work with an ophthalmologist and was able to basically look at images of his clinical cases and actually kind of basically make a trivia for people to understand kind of how glaucoma may appear versus a fungal infection versus somebody exposed to diabetes. So that was kind of interesting for me to look at those images and understand those changes. And then as I got in college, I did studies in biochemistry and yeast. And then when I knew I wanted to be a veterinarian and get involved in research there, I got involved more with animal model design.
Now, and with the blood, say I'm still on the blood-brain barrier, the blood-brain barrier thing. It's because that's what I think you kind of stopped me with. And when I started seeing that, looking at those pictures and thinking about it, I mean, I've known people who's had like brain tumors and things like that. And so how do you morph into, and it seems like, is that basically all at this point your lab mostly focuses on now, is how things cross that barrier in order to treat cancer then?
Yeah, so the central focus of my laboratory is to understand how we can improve therapies through the blood-brain barrier, primarily in metastatic lung cancer and also in traumatic brain injury. So right now, and that's simply just because of the manpower I have, there is really no limit of the diseases that we can look at. Any disease that impacts the brain impacts the blood-brain barrier.
I had a question about when you were talking about, is it androcytes?
Astrocytes?
Astrocytes. And how are those only found in the brain? It looked like we were sort of looking at a cell or something. And then are those in both like cells in the brain, but then also other cells, or is that like a feature you would see throughout the body?
Yeah, so astrocytes are part of an important family of cells called glia. We find glia throughout the brain or neuroparenchyma. So really important cells that can be protective, but also when they get too numerous or don't do their jobs, then they can actually cause damage to the brain. So they're what we call a double-edged sword. So they can be when they're doing the right thing, they control vascular flow and they protect the brain. When you get too many of them, it's a big problem. Or like I said, if they're not doing their job. Now, other cells that are found throughout the body that are part of the blood-brain barrier would be endothelial cells and also the pericytes that I mentioned. However, the endothelial cells and pericytes are usually found about 1-to-1 in the brain. However, in heart or in muscle, you may only find one pericyte for 3 or 4 endothelial cells.
Oh. And what advantage does that have?
So again, the blood-brain barrier, its key feature is that it is largely impermeable. So all of those pericytes help to tighten that barrier. Whereas in the muscle, in lung or liver, in heart, you can have basically a more permeable vasculature, and that would allow proteins or other components to penetrate into the organ parenchyma.
Okay. Wow, that's holy moly. That's more than I knew about biology in a long time.
Yeah.
But I like the fact that you started off by asking questions.
Yes.
I mean, you say thinking about being a reporter and realize asking questions, and because we tell students that a scientist is just someone with questions trying to figure out the answers to them.
Oh yeah, I mean, one of the things that's the greatest, really one of the greatest things about my job is that you never run out of questions to ask and run out of ways to approach things. Because even if, in the experiment I showed you, if sometimes, we may do an experiment for the 10,000th time, and for whatever reason it doesn't work, so we have to go back and say, well, why didn't it work? What changed? What do we need to change to actually make it work? And then also when we think about other diseases, I said that we only do right now metastatic lung cancer and TBI, but even if in current events, like if we're going to eventually evaluate what's occurring in the blood-brain barrier in coronavirus, in other models as well. So it's really important to really just not stop asking questions. And I know, oftentimes it's easy, and I did this all the time too, as a student, is to say, that's a stupid question. I shouldn't ask that question. But, there really is no question that shouldn't be asked. I mean, it may be that it could have been something that perhaps was overlooked in the reading, but, you just need to go back and reread it again, and that's okay. And then I think if you go back and reread something that they've had a question about, you get a new insight and a new perspective on that. And maybe there's something that could have been missed in a diagram. You say, hey, let me go back and look at this text with a diagram. But learning how to be curious is definitely something that allows scientists to be really productive in their careers.
I love your comments on that. And do you have any tips for kids? I think sometimes kids struggle a little bit. They're asked so many questions, but then when we ask them to think of questions, they struggle like, well, I don't know. It just feels very overwhelming. Do you have any tips for kids to help them develop those questioning skills?
Yeah, I mean, even I think, and when I think of questions, I try to write them down, even about just, sometimes I look at the clouds, and sometimes you see the weatherman that's telling you how if the cloudy front is coming in or going. But then, do you ever wonder why clouds have one particular shape in one area of the sky and a different shape in the other area of the sky, even though the weather is the same in both areas? So I think of questions like those, and I don't know the answer to that. Like, why in some areas use perfectly blue, and then you have one cloud that looks like a dinosaur, and then one cloud that looks like the Empire State Building on the same day.
I like
I
that.
like that. I do. I like that. Just all of them, honoring all
Yeah.
of those questions.
So I just write questions down, save them in the notes of my phone. So I know you guys have your phone, so just
Yeah.
save them in the notes portion. Then when you happen to have a chance to meet a weatherman or to meet somebody or to tweet them or reach out to them, send them those questions. They'll love
Oh, I love that. Yeah, record it right in your phone. That's a great.
it.
I was gonna say, as you were talking, I had so many questions come up with myself.
Yeah.
Thinking the blood-brain barrier, are you mainly looking at what the different medicines? Is that mainly what you're researching? How different medicines will react to that? And so,
So
'cause
great
once you said
questions.
COVID, once you said like the COVID thing, I thought, oh, well, I didn't think about the fact that what its interactions would be there.
Mhm. Yeah, so, well, because we don't know a lot about the biological patterns of the blood-brain barrier, so what my group is focused on right now is to understand those biological patterns and understand the pathology first. And then I have a great opportunity here at Purdue to work with chemists who are developing compounds and drugs to use on different diseases.
Okay.
So
I
yes,
thought
so
you were
as a veterinarian, my focus is on the animals and the biology, but as a researcher, I'm able to find others to help me to then target the diseases. So that's another really fun thing about being a researcher is that even if you don't have the area of expertise, so I cannot, in my skill set, create a drug in the laboratory. But that doesn't mean that I can't reach out again and ask questions and get to meet other friends and colleagues who can help me in my group and compounds that they've developed and use in our model system. So they may not have expertise in working with animals and working in model systems, but I do have a compound that may be effective. So it's really important to work as a team to solve these problems.
Oh, perfect. And I love that. I love that you bring that up too, the collaboration point. And anything we want to do, we need to collaborate with people who know more than us in different areas, and it takes all of that knowledge to get things done. And I love that you point that out.
And I also want to say that you know I've certainly tried to present an accurate view of my story, but sometimes I have team members, just like I'm sure you guys sometimes have lab members, where it's simply not a good fit, and that's okay because there's a lot of people on Earth and there's a lot of people around that sometimes you just got to find a good fit. So it may not be the right fit the first time, but usually in subsequent times we can find a good way to work together to solve problems.
Yes, indeed.
Yeah.
Yes, indeed. Well, thank you for your time. We really appreciate this.
Yes.
Yeah, thank you so much. I really enjoyed it.